To start this lab, my group added 4 sterile oats to a petri dish filled with agar. We then introduced 4 cultures of slime mold and placed them on the oats, which was their food source in the experiment. My group's hypothesis was that the slime mold would consume the oats and connect each separate culture throughout the maze, which was created to challenge any outward growth of the slime mold. As you can see in this picture, one error which we did not account for was the movement and separation of certain cultures from an oat. This prevented the cultures in the middle and bottom left of this picture from having an immediate food source. At this point in the experiment, you can see that the cultures have not grown outward. One other important thing to note is that the cultures change from a bright yellow to a more brown hue, which hints that the cultures have begun to die without a food source. In this photo, there also appears to be condensation, which we hypothesize is attributed to respiration occurring. This photo concludes the control experiment. Our conclusion was that outward growth did not occur. However, simple errors like movement could have affected this outcome. For the next stage, we added 2 independent variables to the experiment, 0.1 molar hydrochloric acid and 0.1 molar sodium hydroxide. The dependent variable in this stage remained outward growth. Here we can see that in the top left, the acid has led to the formation of a white mold. In the bottom, however, both the culture and the oat have been broken down into a mush by the base. In conclusion, our hypothesis was not supported. The mold was not able to grow. However, while the acid and base did not lead to the growth of the slime mold, they did act differently than the control, with a stiff white mold growing on the acidic oat. We did, however, attempt to transplant a partially grown slime mold after the experiment. As you can see, a different mold grew along with a small slime mold. This may have been due to human errors such as breath or incorrect sterilization procedures.